Thanks for being here. I'm Harrington. I'm in Todd Reader's lab at San Diego State, and I'm going to be talking today about phylogeography within Crotalus ruber using Rattlesnake data. So Crotalus ruber is a red diamond rattlesnake, is a large rattlesnake found throughout Baja, California, up into Southern California, with the range shown here in the dark gray. Traditionally, based on morphology, there were two subspecies recognized. Crotalus ruber leucosensis, shown here in the south of Baja, California, and Crotalus ruber in the north of the mainland of Baja, California, shown here in dark gray. And the boundary between these two subspecies, which were traditionally recognized based on coloration and scalation, was right about here near the Mexican town of Laredo. And at the boundary here, there is this putative intergrade zone where morphological intermediates were found, and there was a process to use intergrades between these northern and southern populations, or subspecies. However, some previous work using mitochondrial DNA to look at divergences across the mainland populations of Crotalus ruber showed very low intraspecific mitochondrial divergence from extreme northern to extreme southern Baja, California of only about 0.3%. And this contrasts with other mitochondrial divergences in western rattlesnake species of up to about 6% divergences within the Crotalus michelli complex, although some of these have been recently elevated to species status, and 4% divergences found within Crotalus cerastes, the side one here. So despite the fact that Crotalus ruber has not been shown to have strong mitochondrial breaks across the mainland peninsula of Baja, many other squamates do show major genetic breaks across the peninsula, some that are listed here. And these breaks are generally found at one or more of three spatially concordant areas throughout Baja, California. The most common being this mid-peninsular break in the middle of the peninsula, and then other less common breaks occurring at Laredo, which is also that boundary between the subspecies of Crotalus ruber, and also this boundary at the Isthmus of La Paz. So the objectives of my study were to use a next-generation sequencing approach to assess the population genetic structure within Crotalus ruber, and see if using a genomic perspective that has a greater power to detect structure would detect structure that was not detectable using mitochondrial data alone. And so I chose to use a RAD-seq approach, which is, as many people probably know, a genome-wide reduced representation library method. So then the objectives were to use this RAD-seq data set for Crotalus ruber to investigate the population structure and identify if there are breaks at any of those regions of Baja, and investigate the phylogeographic history to identify if breaks are concordant with the breaks found in other taxa, and then also to perform phylogeographic model selection to determine the phylogeographic history of this species across the peninsula. To do this, I obtained 35 Crotalus ruber tissues from the San Diego Natural History Museum and Muabe collections in Mexico. Sampling localities are shown as the black dots here within the range of Crotalus ruber in the gray. So I have good sampling across the mainland range of the species. I then used a ddRAD approach and sequenced libraries on Illumina HiSeq using the 100 base pair single end reads, and I used the PyRAD bioinformatics pipeline for data processing. I obtained a total of about 4.5 million reads, which then resulted, let's skip down just this last part here, in about 1,500 loci when using a matrix completeness threshold of about 75%, for a total of 1,800 or so variable sites and 946 unlinked SNPs. I took several approaches to analyze the structure and phylogeographic history of Crotalus ruber. First, I performed a couple of concatenated analyses using RAXML. RAXML, of course, assumes a bifurcating topology, and so is not necessarily the most appropriate for these kind of population level questions, but serves as a good first pass to see how individuals cluster in the phylogeny. And I also estimated the phylogenetic network using SplitsTree, which does not force this bifurcating topology. Next up, I used model-based population clustering methods, structure, and the similar maximum likelihood admixture to identify population membership and admixture of individuals. I also used non-parametric clustering approaches, PCA and spatial PCA, and also estimated migration, divergence, and population size parameters using GFOX, which is a Bayesian method that's somewhat similar to the program IM and IMA, and then it estimates these divergence times, population sizes, and migration rates among populations. And finally, I performed phylogeographic model selection using the program FastSimCol2, which uses site frequency spectra and a simulation approach to estimate demographic model parameters for any arbitrarily complex model that can be specified, and also approximates the likelihood of these models using simulation. And with these likelihoods that are obtained, models can then be compared and selected using AIC as in other model selection scenarios. The three main models that I tested included a model of divergence with no migration, shown as the cartoon on the left here, with populations are isolated as soon as they diverge with no migration between populations. I then tested a model of divergence with migration, or basic isolated migration model. The populations diverge, but then migration continues between these populations after divergence. And finally, I tested a model of divergence with secondary contact here, where the populations diverge, and for some period of time, there is no gene flow between the populations, and then afterwards, gene flow begins between these two populations again. So the Rattlesnake and splits tree results were both highly concordant. The deepest split in this Rattlesnake tree here is between a cluster of southern individuals and northern individuals. And the boundary between these two clusters is shown here, and this is right about at Loretto, so concordant with the boundary between subspecies and also this Loretto break found in other taxa. The splits tree also clustered southern individuals and northern individuals, with some other individuals falling out as somewhat intermediate, suggesting the possibility that these are intergrades. And the starred individuals here and in the Rattlesnake tree are actually individuals that are identified as intergrades by structure and admixture, which I'll get into next. So structure and admixture both produced highly concordant results that were nearly identical, so I'm only going to present the results of admixture here. Both programs favored a K of 2 as the optimal number of populations within Corollas ruber. And they both identified a southern population, shown here in the blue, and a northern population, shown in the yellow, also shown on the map, with several individuals that were shown as intergrades or admixed between these. So those are the groups that are shown here. And you can see that again on the map here, the break between these populations occurs at this Loretto break, where individuals that are further from this break are shown as less admixed than the individuals that are closer. PCA and spatial PCA also produced highly concordant results. PC1 of the PCA largely differentiated southern individuals on the left here from northern individuals, with the same starred individuals being those that are admixed as detected by structure and admixture, shown as intermediate on PC1 here. And then again, in the spatial PCA, here individuals are plotted according to their geography. And here we have a strong differentiation between southern samples and northern individuals, with these admixed individuals coming out intermediate once again. So using GFOX, I estimated the population sizes divergence between populations and migration rates. And I showed that the northern population is considerably smaller than the southern population, only about half the size. The divergence time is estimated to be about 200,000 years between the divergence between these northern and southern populations. And the migration rates are estimated to be highly asymmetric between these two populations, where the migration rate from south to north is considerably higher than the migration rate from north to south. Using FastSimCol, I found that the secondary contact model provided the best fit. And again, this is a model where the northern and southern populations were isolated for some period of time without migration, and then after a period of time, came back into contact, and migration is now going on between these populations. The estimated migration parameters were similar to those estimated from GFOX, again showing this highly asymmetric gene flow with more gene flow from north to south than vice versa. And for a divergence time of about 200,000 years on that estimate from GFOX, the secondary contact is estimated to have begun at about 40,000 years ago. So all of these analyses strongly supported two populations that were highly congruent among analyses, with this break occurring here at Loretto. These populations are concordant with the previous subspecific taxonomy, with Corollas ruber in the south and Corollas ruber in the north. And this break is also concordant with a break that's found in other taxa, including the lizards Calosaurus and Urosaurus magnicaudus, although both of these lizards also show additional breaks across the peninsula, not only this Loretto break as in Corollas ruber. And so using FastSymCol, I showed that the model of secondary contact is the best fit to these data. And FastSymCol and GFOX both support a highly asymmetric gene flow between these two populations, with much more gene flow from south to north than north to south. And so in addition to this higher gene flow between populations to the north, there's also some evidence of a northern range expansion within the northern population. So looking at the Rattlesnake tree, these northernmost samples are the most highly nested within the northern population, whereas the southernmost samples are more basal. And in addition, the GFOX and FastSymCol analyses estimated much smaller population sizes for this northern population, despite the fact that the northern population has a much larger geographic range. So together these suggest the possibility of a northern range expansion within this northern population. So overall, these RAD-seq data identify population structure within Perlis River that was not detectable using previous mitochondrial data alone. And GFOX and FastSymCol suggest a complex phylogeographic history for the species that includes secondary contact and asymmetric gene flow between populations. Although the causes of the divergence between these northern and southern populations are still somewhat unclear, although the timing of the divergence at 200,000 years ago and the secondary contact at about 40,000 years ago are both well within the Pleistocene, suggesting the possibility of climatic fluctuations as the cause of these divergences. With that, I'd like to thank my advisor who helped me out with this and my funding sources, and I should have a couple minutes for questions.